Yeast is an amazing creature. It has the ability to survive in both aerobic and anaerobic conditions.
What does that mean?
Yeast undergoes cellular respiration, which means that regardless of whether oxygen is present or not, it can still produce enough of it to sustain itself.
But why are we bothering you about yeast? And what does this have to do with outer space? And why are we asking you all of these questions?
Well, experiment question. Could yeast growing on other planets produce enough oxygen to sustain human life? Could this be how the first organisms originated in an oxygenated environment?
If yeast cells do proliferate in outer space, they could provide enough oxygen to eventually create an environment that mimics Earth's atmosphere for human survival in approximately 1 million years, which would be found by extrapolating the results of a 4-week experiment.
Have a yeast cell plate and habitat 2 in an anaerobic environment to mimic all the specifications of outer space, such as zero gravity and a vacuum, to see how it proliferates and to measure the oxygen production of the yeast. Have a control on Earth and normal conditions to compare. Monitor the setup every day for a few minutes. This should not take more than 5 minutes. Record signs of colony growth in each habitat for 4 weeks. Then, record the amount of oxygen production each day. Compare the results to those in a control on Earth. At the end, use data to calculate how much oxygen would be created in 1 million.
We expect that the yeast grown in outer space will produce enough oxygen in 1 million years to sustain human life similar to that on Earth. This is completely safe and bio-teacher approved.
I'm Allie Lane, age 16.
And I'm Sonali Joshi, age 16. And this is our YouTube Space Lab experiment.